So our final macromolecule that we're going to talk about are the proteins. Proteins are the building blocks of life, and you can see here lots of examples of where we get our proteins in our diet. But proteins primarily include the enzymes, as well as lots of transport proteins. Shown here is hemoglobin, which transports oxygen, as well as structural proteins. These include things like keratin and collagen, which provide structure to our hair in the case of keratin and our bones in the case of collagen. Proteins primarily are involved in enzymatic reactions, transport, and providing structure. Here you can see the monomer for proteins, which are amino acids. So amino acids are the monomer, polypeptides are the polymer, and polypeptides are composed of amino acids that are linked by peptide bonds. So our monomer is an amino acid, our polymer is a polypeptide, and the linkage between monomers is the peptide bonds. And there are 4 major classes of amino acids, and I'm going to ask you to learn 3 things about each amino acid. So you don't have to know the structures for all the amino acids. We'll save that for biochemistry. But for all 20 amino acids, you should know for that amino acid, what is its 3-letter symbol, what is its 1-letter symbol, and what class does it belong to. And to understand what class it belongs to, we need to talk a little about the structure of an amino acid. All amino acids have this central carbon known as the alpha carbon. The alpha carbon is surrounded by 4 different groups. First, you have this functional group, which you should know is the amino group. The amino group is positively charged at physiological pH. The second functional group you see here is the carboxyl group. The carboxyl group is, of course, negatively charged at physiological pH. Here you have a hydrogen, and together these 3 groups, shown here in purple for all of these amino acids, you'll notice that they're identical. So this is the backbone of the amino acid. The part that varies is the side chain, which is also known as the R group of the amino acids. So the R group is what gives each amino acid its character. And you'll notice here, for this group of amino acids, all of the R groups are composed of carbons and hydrogens for the most part. We see the occasional nitrogen, but it's not really in much of a position to be reactive, and the occasional sulfur, but once again, it's sort of blocked by these carbons. So in this case, these amino acids are primarily composed of non-polar covalent bonds, and we group these amino acids as non-polar amino acids. These amino acids, their R groups tend to form non-covalent van der Waals interactions because they're non-polar. Our second group of amino acids are the polar amino acids. You'll notice, once again, our backbone is the same. So we have the alpha carbon, the amino group, and the carboxyl group, and our hydrogen. But here, these R groups, shown in green, tend to have more reactive groups. They have these hydroxyl groups. We see lots of hydroxyl groups. We have a thiol group here. These are actually known as amide groups. They have a carbonyl right next to an amino group. But all of these tend to form hydrogen bonds. So these are what's known as the polar amino acids because they have polar R groups. Once again, here you can see the 3-letter designations and the single-letter designations for each amino acid, and these tend to be hydrophilic. And so our last 2 groups of amino acids are the acidic amino acids and the basic amino acids. Once again, our backbone is the same, but the acidic amino acids are negatively charged at physiological pH, whereas the basic amino acids are positively charged at physiological pH. So these guys would tend to form ionic bonds with one another in the context of a protein. So these are very hydrophilic because they're charged. Now, here is your exciting bonus opportunity for sticking it out and watching this video before Friday. I told you were going to get something out of this. Each one of you, in order to help you learn the amino acids, the 3 things we need to know for each amino acid. It's single letter designation, it's 3-letter designation, and what class it belongs to. Polar, non-polar, acidic, or basic. You should make flashcards showing the amino acid and each one of those 3 things to practice with, because you're going to need to know these for your first exam. So if you go ahead and make your flashcards and then take a selfie of yourself with your flashcards, hopefully somewhere fun just to entertain your professor, and if you email it to me before Friday at 8 a.m., so before we have class, then you'll get 1 bonus point. So make sure to take advantage of your awesome bonus opportunity and to make your flashcards and start learning those amino acids, because you need to know it by the first exam. Moving on from those amino acids, how do amino acids assemble into proteins? We already know that amino acids are going to link to one another through a dehydration synthesis reaction. How that occurs in this case is we remove this OH and this hydrogen, making a water molecule, and then through this dehydration synthesis reaction, we'll form a bond between this carbonyl carbon now and this nitrogen, and that's what's known as a peptide bond. And much like nucleic acids, amino acids only assemble in a linear fashion. So because of that, that means that amino acids and polypeptides also have directionality. One end of a polypeptide is always going to have this amino group, and so it's known as the amino terminus or the N terminus. And the other end is always going to have a carboxyl group. So it's known as the carboxy or carboxyl end or C terminus. So the peptide bond is between the carbon and the nitrogen, and we have the N terminus and the C terminus. We just talked about that proteins assemble in a linear manner. Here you can see we have glycine, then proline, threonine, then glycine, and so on and so forth. This is what's known as the primary structure. This is the linear order of amino acids, and it's usually written from N terminus to C terminus. The second level of protein structure is the secondary structure, and there are only 2 major types of secondary structure. That's the alpha helix, shown here, and the beta pleated sheet. These 2 different secondary structures are very different from one another, but they both share some common characteristics. First, a secondary structure is stabilized by hydrogen bonding within the backbone. So it's actually hydrogen bonding between that carboxyl oxygen and then the hydrogen that's attached to the nitrogen. And those hydrogen bonds in the alpha helix occur between amino acids that are about 4 amino acids apart. So 1 turn away on the helix ends up being about 4 amino acids apart. Whereas in the beta pleated sheet, the hydrogen bonding occurs between different beta strands. So these are different strands that could be really distant from one another within the primary sequence of the protein, but in 3D space, they end up being really close to one another. So the hydrogen bonding occurs between beta strands in the beta sheet. Another thing that these have in common is that both the alpha helix and the beta sheet cover the backbone, so they sort of hide the backbone and then face these R groups out to protect the backbone. So in the alpha helix, the backbone forms the core of the helix, and then these R groups point out into space around the outside of the helix, kind of like the spokes of a wheel. In the beta pleated sheet, if you think of this sheet as a sandwich, then the R groups alternate pointing up and down out of the plane of the sheet. So you can kind of imagine that if a beta pleated sheet were a peanut butter and jelly sandwich, then the peanut butter and jelly would be in the middle, that would be the backbone, and the 2 pieces of bread would be the R groups. So it hides that backbone within the sandwich. Another thing that I should point out is that beta-pleated sheets can come in two flavors. So if these beta strands run in opposite directions, they're what's known as an anti-parallel beta-pleated sheet. Whereas if these strands were running in the same direction from N terminus to C terminus, that's what's known as a parallel beta-pleated sheet. Our third level of protein structure is tertiary structure. And we said that secondary structure depended on interactions, hydrogen bonding between the backbone of the amino acids. In contrast, tertiary structure is how the 3D protein begins to fold. And now we sort of tucked that backbone away within the secondary structure. So tertiary structure is stabilized by interactions between the R groups. You can imagine that polar amino acids may form hydrogen bonds with one another, whereas more hydrophobic or non-polar amino acids are more likely to form van der Waals interactions. Acidic and basic amino acids, and there's actually one type of covalent bond that can form. Cysteines, shown here, if you remember, cysteine had a thiol functional group. So cysteines can form covalent disulfide bonds or disulfide bridges, and these also stabilize tertiary structure. Just to review, tertiary structure, or the 3D fold of a protein shown here, is stabilized by interactions between the R groups. These include the non-covalent interactions as well as covalent disulfide bridges. So let's think for a minute. So where are hydrophobic amino acids most likely to be found? You know that hydrophobic amino acids are most likely to be found away from water. So because of that, hydrophobic amino acids are most likely to be found within the interior of a protein, or alternatively, if you have something like a membrane protein that's embedded within the membrane, then they might be exposed on the surface to allow the protein to interact with those fatty acids. The final level of protein structure is quaternary structure. Now, not all proteins have quaternary structure because quaternary structure occurs when a protein is composed of multiple polypeptides. You can see several examples here. For example, collagen is this triple helix, so we have 3 different polypeptides. Hemoglobin, shown here, is composed of 4 different polypeptides, so 2 alpha and 2 beta subunits. And transthyretin, shown here, is composed of 4 identical polypeptides. And just like tertiary structure, this is stabilized by interactions between the R groups. It just occurs when proteins are made up of more than one polypeptide. Just want to review for a minute. So our 4 levels of protein structure were primary structure, which is the linear order of the amino acids. Secondary structure, which is the alpha helix and beta sheet, and it's stabilized by hydrogen bonding within the backbone. Tertiary structure, which is the 3D fold of the protein, which is stabilized by interactions between the R groups. And quaternary structure, which not all proteins have, but for proteins comprised of multiple polypeptides, it's stabilized once again by interactions between the R groups. So finally, I just want to mention why we're focusing so much on protein structure. And that's because protein structure really matters for human disease. I think many of you have probably heard of sickle cell disease. This is a disease that is really prevalent among African Americans, and that's because it was prevalent in regions of the world where malaria was endemic. So these include parts of Africa as well as parts of the Mediterranean. So there are also Mediterranean populations that have really high levels of sickle cell disease. So sickle cell disease occurs when we have a mutation in the beta subunit of hemoglobin. So normal hemoglobin, you can see represented here, folds like this. Whereas in sickle cell hemoglobin, you can see here that this glutamic acid, which is a negatively charged amino acid, is changed to valine. So valine is a hydrophobic amino acid. So now, instead of having a negatively charged amino acid hanging out on the surface where it's happy to interact with water, we have this hydrophobic amino acid, which we know doesn't like to hang out with water. Because of that, during a sickle cell crisis, usually when oxygen levels are low, this hydrophobic piece that is exposed can cause the hemoglobin to self-assemble into a polymer, essentially. And that polymer deforms the shape of the red blood cell, making it sickle-shaped, hence the name of the disease. And these sickle-shaped red blood cells and can block blood flow through the capillaries. And this is what's known as a crisis event, and it's really, really painful for people who have this disorder. And the treatment is usually to give them oxygen, because oxygenating the red blood cells can help to disassemble these polymers. But this can be a really painful event, and because of this, many people with sickle cell disease have a much harder time doing athletics, because they're more likely to deprive their hemoglobin of oxygen when they're working their muscles really hard. So this shows you why those amino acids are so important, and understanding how those R groups interact with each other can be very important, because ultimately one single amino acid change from a negatively charged amino acid to a hydrophobic amino acid can lead to this devastating disorder. And one interesting tidbit about this disease, why it's so prevalent in these populations that came about where malaria was endemic, is people who are heterozygous for the sickle cell trait, meaning that they have one disease copy of the gene and one normal copy of the gene, are actually resistant to malaria. And that's because the turnover of their red blood cells is much faster. It occurs much more rapidly. So because of that, the malaria parasite, which grows within the red blood cells, is less likely to take hold, so they tend to be resistant. So that's why that trait is more prevalent in populations that originated from the Mediterranean region or from Africa. So I just want to point out that this protein folding that we've talked about, so forming this tertiary and quaternary structure, is reversible. So we can denature proteins by breaking the bonds between R groups, and we can renature proteins by then reforming those bonds between R groups. Denaturation can occur by treating proteins with heat, which of course can break bonds, by salt, which could break ionic bonds, or by changing the pH, which can break either ionic bonds, it can definitely break hydrogen bonds. By treating with different conditions that we can break these bonds between R groups within the proteins, we can denature the protein or unfold it. And that's going to become very important for lab in the next few weeks. So I wanted to point that out ahead of time. And just an interesting little tidbit, one thing that we know now, we actually can't predict protein structure just based on primary structure, just based on that linear order of amino acids. And it's kind of the holy grail of biochemistry. If quantum computing ever gets fast enough to try out lots of different configurations of a protein and find the most stable, that will probably be Nobel Prize worthy, because predicting protein structure based on the linear order of amino acids would actually allow us to design our own proteins and enzymes, which could revolutionize biotechnology. And if any of you guys want to try your own protein folding, there's this game called Fold It. It's actually a game where you can fold proteins on your own based on these different characteristics. So you may, if you want to learn more about protein folding, you may want to try this game. This would also be a good way to practice learning about those different R groups and amino acid structure. And for right now, it actually seems that humans sort of working in the cloud can predict protein folding potentially better than computers. So this is an interesting game for you to check out. And that is going to be the end of this video. So don't forget to take your selfies with your flashcards and send them to me by email before class on Friday. And we'll be doing some clickers based on your notes. And feel free to bring your notes on this video. We'll be doing that on Friday, and some activities using what you've learned. So I will see you then.